going to share with us. We also will have some time for you to ask questions. So during him sharing his research, feel free to drop questions into the chat. But if you have questions at the end that you want to ask, that is a time that we're saving for y'all. And then our team has done some research for you as science teachers, as science educators, or just in the science space to say, hey, this is how you can take this really cool research and bring it into your classroom and make it applicable and how it can kind of fit into your curriculum. And then we believe in making sure that you have the resources to be able to take action once you get back in your classroom, whether that be next week or next month. And so we have some resources for you and some information about our next event. So who we are, my name is Hannah Wedger. I am a part of a team with Sarah Nerswick and Angela Galata. We're all former teachers. We have over 30, close to 40 years of experience in the classroom. And we really work in the NGSS space and supporting teachers and professional development. Um, and we're excited to be here to be kind of the go-between between the research that Marshall is sharing and how we bring that into your classroom, like we said. So we represent and are here on behalf of the Food and Agriculture Center for Science Education, and really thinking about providing this series. So this is the first one of a series of virtual forums where we are helping to connect food and agriculture researchers with science educators, which is you. And so our goal is one for y'all to get access to high quality science phenomena and to kind of figure out how does that work into our curriculum. And then the other piece is how do we connect researchers who are doing really, really cool things and allow you to know like this is how you can bring it into your classroom and really provide this space where as uh, as teachers, as curriculum writers, and as folks who are doing the research, we can kind of have this community of building really high quality instructional materials and experiences for our students. So, without any further ado, I am super excited to welcome Dr. Marshall Wedger. He is a distinguished researcher at WashU in St. Louis, and he really brings a wealth of knowledge to this forum here today. His background is rooted in rigorous I would say inquiry and just a commitment to advancing biological sciences. Marshall does research on weedy rice, which y'all are going to be so excited about this. You may have never heard of weedy rice before, and at the end of it, I am sure you're going to be like, whoa, this is really cool. And so he's just really dedicated to kind of uh, unraveling the complexities of the work that he does and really, I would say, working to inspire the next generation of scientists. So I am going to hand it on over to him so he can share more. Marshall, go ahead and take it away.
Great. Thank you so much. Okay. Thank you so much, Hannah, for the introduction. Hello, everybody. My name is Marshall Ledger. I study Weedy Rice here at Washington University. My email is right here down at the bottom. Please write it down and feel free to email me any questions you have. I love talking science with anyone who can talk back to me. Okay, so first I want to just very quickly say who am I, why am I talking today, where did I start, where did I get to, what am I doing right now? So I grew up in northern Minnesota, where I went to college in Duluth, and I worked with a wonderful researcher named Dr. Brianna Gross up there. We studied the adaptation of apple trees to the really cold climates in northern Minnesota. And it was really like through her lab, through her mentoring that really got me into science. She also then pushed me to do a PhD. So right after I finished my bachelor's degree in biology, I moved right into a PhD at Washington University in St. Louis. Um, that while I was there, I met two wonderful researchers. First, here's Dr. Nilda Roma Burgos at the University of Arkansas in Fayetteville, and then Dr. Kenneth Olson here in the back here. This is my primary mentor at WashU. And these two people are incredibly intelligent and wonderful mentors, and I really owe a lot to these two people in particular for all of the research you're about to see. So, like I mentioned, I'm in the Olson lab, and so what is it that the Olson lab does? We study the genetics of adaptation in plants. That's sort of the broad umbrella term for everything that we do in the lab. I encourage you to visit our lab website, olson-lab.org, to see all the things that we do, but I've put up just a smattering of things here. All the way from these are all this is my cohort. These are all the graduate students that I was a graduate student with. This is David Goad. He studied salt tolerance in seashore paspalum. This is a grass that you can actually water with seawater. And so golf courses that are on the coast use this grass so that they can actually water and irrigate with seawater. It's incredible. Sarah Wright, and another incredible scientist here. She studied climatic gradients all the way from northern Minnesota to southern Florida and how white clover adapted to those crazy climatic gradients. Jordan Brock here, he studied the biofuel production in camelina. This is an oil that you can literally like extract from the plant and put into an airplane, and essentially the airplane will work. So just really cool stuff that our lab is doing, and any one of these people could be giving this talk today. But I'm giving the talk, so who am I and what do I do? Well, I study a species called weedy rice. I specifically study the kind that you find down in the southern United States. We grow rice sort of in two areas here in the U.S. The southern U.S., which includes like Arkansas, Missouri, Louisiana, Texas a little bit, and then a second one over in California. I don't work as much in California. I work mostly in the southern U.S. Weedy rice is what we call a weedy conspecific. That means it's the same species as rice. It's actually a feral strain of cultivated rice. And what this means is that weedy rice essentially re-evolves in every world region where we cultivate rice, all the way from the United States to Colombia, Italy, France, the East Asia countries, China, Malaysia, Thailand. All of these places have their own unique independent evolutions of weedy rice. So I am sort of in charge of the US kind. And it's wildly competitive. It competes for light, it competes for space, competes for nutrients, and it's terribly devastating economically. Um, Farmers are known to just like abandon fields if the weedy rice gets too bad. Um, Up here in this picture here, everything you're looking at right here is weedy rice. This is a field that has been abandoned because the weedy rice is so bad. This particular little patch here um, is weedy rice that grew so fast that it lodged, which means that the stem didn't have time to get strong enough to support the plant. So it lodged, it fell over, and it killed all the cultivated rice underneath it. Those are all losses for the farmer, right? This picture down here at the bottom, this is actually one of my favorite pictures I've taken of a pretty infested field. You can see actually like two canopies, right? This bottom canopy here, this is cultivated rice. This is what we actually want. And this top canopy up here, this is weedy rice, right? And with the weedy rice being on top, you can just see, right? All the light resources that are taken, all of the soil resources that might be taken, all the space that might be taken up, and all of this is really terrible for cultivated rice, um, and for farmers that have to try to deal with this, right? The presence of weedy rice actually lowers quality of cultivated rice. It lowers yield. It lowers anything that a farmer might use to try to sell their rice. So it's really economically devastating. Okay, so in order to sort of like begin to understand what weedy rice is and what it does, we have to first actually learn a little bit about cultivated rice. So I'm showing here what the taxonomy is for Asian cultivated rice. This is all the rice you would buy at the grocery store. Up here in the gray, this is Oryza rufipogon. This is the wild ancestor to all Asian rice. Through a pretty hazy domestication process that I'm happy to talk about later, we come up with two sort of subgroups of Asian cultivated rice in the dark blue circles. The one on the left here is the Indica group. The one on the right here is the Japonica group. And in the United States, we grow primarily tropical Japonica varieties. Um, that's changing a little bit since supply chain disruptions because of COVID, but historically it's been 100% tropical japonica. And over here on the right, this is a picture of a really well-behaved tropical japonica cultivated plant. You can see it doesn't really interact with its neighbors much for space. All of the seed heads are at about the same height, making harvest really, really easy. And you can see that like the seed heads are full and they're on the plant, which is important if you want to collect the rice. Here in the United States, we have actually two strains of Weedy rice. I've been talking about it as a sort of monolith, but we have two independently evolved strains of Weedy rice, and they're actually from these varietal groups, Indica and Aus. So derived from Indica-like plants, we have straw hull plants, straw hull Weedy rice. Straw hull Weedy rice is kind of like a crop mimic. It takes a mimicry approach. It looks a lot like cultivated rice until you break open the seed coat, the hull, and you see these really bright red pericarbs. That's why it has if you talk to farmers, they call it red rice, and it's because of these red pericarbs. Black Hole Ond, on the other hand, is just takes a competitive approach. It grows taller, it grows faster, it grows more aggressively, it takes up more space. And this is derived from the Aus varietal group. So as a quick recap, tropical japonica is what we grow in the United States. Straw hull rice, Weedy rice, and black hull Ond Weedy rice are our two varieties that at least historically grew in U.S. rice fields. All of that work, all of that knowledge was from people before my time, and I want to walk you through it because it'll become important later. What we're looking at right here is called a structure graph, and these are what we use to sort of assign ancestry. We do a lot with structure graphs, but one thing you can do is you can assign ancestry. And it looks kind of complicated, but it's really not. So each vertical bar is one sample that we went and collected from the field. And the degree to which that bar is colored, one color versus a different color, is the degree to which structure, the program that we're using, has assigned that individual to a certain population. So this vertical blue bar is one sample that is very closely related to another vertical blue bar, whereas this pink bar is sort of a different genetic subgroup. Um, then you get samples like this one here, where the bars are kind of like partially one color, partially another color. That's a sample in which structure has decided that it's likely that this individual is actually an admixed individual from two different populations. So to understand this graph, all you need to do is look at the colors. Here I've labeled the US crop this is dark blue, straw hull Weedy rice in pink, and the black hull Ond Weedy rice in green. And the major thing you need to take from this is that they're pretty much solid colors. There's very little, these are samples collected from the 1990s. Um, there's very little genes moving from one population to another. And that's interesting, right? Because they all grow in the same field. You can walk out into a field and you can do this, and you can have all three strains in your hands, right? So it's weird that they've spent 150 years growing right next to each other, and they very rarely, if ever, outcross. So no evidence of outcrossing for the 150 years worth of history. Um, Times are a changing though, and in the early 2000s, a bunch of mechanistic changes happened to rice agriculture. One of which that we're going to talk about is the introduction of herbicide-resistant clearfield rice. So this is a non-transgenic uh, strain or I'm sorry, cultivar of cultivated rice that was introduced as herbicide resistant, specifically to this imidazolinone class of herbicides. Um, like I said, this is non-transgenic, so breeders were just growing up a bunch of rice, hitting it with herbicides, and finally one of them survived. It was a random mutation that they brought into a named strain and commercialized. Uh, they found resistance via amino acid replacements in the ALS gene, and there was actually several. There have been several introductions of Clearfield, all with like different mutations leading to resistance. And when I say this is these technologies were highly effective, I really mean it. They cleared weedy rice from the fields, and farmers loved it. There are anecdotal stories in like Colombia where they talk about how they were getting ready to just live with weedy rice until Clearfield came in and saved the day. Like it's farmers love this stuff, and they adopted it very quickly. These data here in the graph end in 2013 at about 40-ish%, but later data that wasn't published in this particular report suggests that at one point about 60% of all rice grown in the southern US was some variety of Clearfield. And that's incredible. Since that peak, though, it's dropped down, and it's now plateaued at around 35% of our fields because of the reason that we're all here today, herbicide resistance. So, what happened and how fast did it happen and what does the timeline look like? So here we are, we're in 2002, the introduction of Clearfield rice. By 2006, farmers had begun anecdotally reporting instances of herbicide-resistant weedy rice. So that's just 4 years before weedy rice starts to become resistant. In 2010, a colleague of mine in Massachusetts went out and did collections and found about 80% resistance. So that's very quick, that's very fast. And then in 2018, I went out and did my own collections, and I found about 96% of our samples to be resistant to the imidazolone class of herbicides. And so that's just 16 years. In just 16 years, we went from 0% resistance to almost 100% resistance. How does that happen so fast? How can we prevent that from happening in the future? And what sort of techniques can we use to mitigate that? Okay, so that's the primary question of this particular avenue of my research. How did this happen so fast? And I'm an evolutionary biologist, so I'm going to answer these questions with evolutionary biology. And here's sort of my outline for the rest of the talk. We're going to talk quickly about what is evolutionary biology. It's kind of this nebulous term that's a little bit scary, so we're just going to talk about it. Next, we're going to dip our toes into evolutionary biology with the Hardy-Weinberg equation. I'm pretty sure most high schools talk about it. I know mine did, so we'll talk about Hardy-Weinberg. Then we'll get back to the weedy rice and we'll solve the mystery. Okay, so what is evolution? What is evolutionary biology? Evolution, very simply, is a change in allele frequency in a population over time. That's it. That's the definition of evolution. A change in allele frequency in a population over time. Just those four things. Um, so really it's not as nebulous as some might think. It's actually a very well-defined thing that we can go and we can make predictions about allele frequency changes in populations over time. Okay, so I'm going to sort of dissect this definition very quickly. The first thing we need to know is the difference between a gene and an allele. A gene is some fragment of DNA that codes for something. In this toy example, I have the blood type gene. You, me, everyone else here, we all have blood types, whatever they may be. They're coded for by this gene, which is just a fragment of DNA. Within that gene, we have what are called alleles. Alleles are variants of that gene. And so, I might have the AB, or I'm sorry, I might have an A allele, someone else might have a B allele or an O allele, right? These different variants, these are what are called alleles. From these two definitions, you can come up with two more definitions, which are allele frequency and genotype frequency. They sound the same, but they're kind of different. The allele frequency is the fraction of alleles in that population. So you, me, everyone else here, we're all diploids. We have two copies of the genome. So we have two alleles per gene. If we went and we counted all those genes and divided by the population, we would get some frequency of that allele. So in this toy example, the A allele is at 27%, the B allele is at 6%, the O allele is at 67%. Since you and I and everyone else here are diploids, we all have two copies, two different, sometimes the same alleles. And if you put those alleles together, you come up with the genotype frequency. Genotype frequencies are important, they're nice, but as evolutionary biologists, we care about allele frequencies. How are allele frequencies changing in a population over time? Okay, so how can we use these ideas to look at Hardy-Weinberg and see how that equation can help us solve these evolutionary questions? The very first thing you need to know about the Hardy-Weinberg equation is that it's a null hypothesis. We're asking um, we're asking to make predictions about evolution, about evolutionary biology. We need to test those against a null hypothesis, and the Hardy-Weinberg equation does that. Very simply, it says if the allele frequencies P and Q are known, then genotype frequencies are given by this equation, P squared plus 2PQ plus Q squared equals 1. And that's what's shown here in this figure over on the right. As P increases and Q decreases, you get these very well predicted patterns of genotype frequencies. Very importantly, if the Hardy-Weinberg equation holds, then the population is not evolving. And from a lot of work, from a lot of people for a lot of years, we know that populations can only not evolve if 5 assumptions are met. And all 5 of those assumptions have to be met in order for populations to not evolve. So, what are those 5 assumptions to the Hardy-Weinberg equation? Well, luckily they fit right on your hand, so they're pretty easy to remember. Very quickly, this is stripped right out of from a TED Ed talk, Five Fingers of Evolution by Paul Anderson. If you haven't seen it, I strongly suggest going to watch it. It's incredible. Okay, so we're going to start on our pinky finger and we're going to move across the hand. The first assumption to Hardy-Weinberg is infinite population sizes. If only, I'm sorry, if you take a subset of a population, uh, the new frequency will change unless the population is infinite. And so if we care about allele frequencies changing, which we do, then the population has to be infinite. Otherwise, you can't subset and come up with the same population. We'll get to it. The next assumption is random mating. And this, I'm sorry, let me back up. So infinite population size, why does that fit on your pinky? It's because your pinky is the smallest finger. And it should remind you that small populations are in a violation of Hardy-Weinberg. Okay, moving on to random mating then. The ring on your ring finger should remind you of mating situations. And random mating is one assumption that goes into Hardy-Weinberg. It has to be true that every individual has the exact same chance of mating with every other individual in your whole population. Any deviation from that is a violation of Hardy-Weinberg. The next assumption is no mutations. Mutations cannot happen. If a new mutation arises, that new mutation is now at some allele frequency, right? And so allele frequencies are changing, populations are evolving. This one is pretty simple. The M that starts middle in your middle finger should remind you of mutations, which also starts with M. Pretty simple. Next is migration. Why, if you are standing there and you say, I want to go over there, with your pointer finger, you point over there, I want to go over there, and you migrate, you're changing allele frequencies. You violated Hardy-Weinberg. Next is no selection, and that fits on your thumb because your thumb is one of the best examples of an adaptive mutation. Opposable thumbs are great. Okay, so all of these assumptions, they might seem random, but they're really not. These are actually just different ways to say the 5 forces of evolution. Everyone knows about natural selection, but that's just one subcomponent of one force of evolution. There are 4 more. There are 5 forces of evolution. Starting with that, we have selection, we have gene flow, we have mutations, mating system, and genetic drift. All 5 of these things are forces for evolutionary biology, for evolution to happen. And just as a primer, I'm gonna go through each one of these with one single slide as there is a PhD in every one of these forces. And so at the end of this, with 5 slides, you might not entirely get it, but hopefully I'll I'll get you somewhere along the way. So again, we're gonna start at the pinky, genetic drift on the little finger. It's pretty obvious that no population is infinite. This assumption for Hardy-Weinberg can never be met. And so populations are always evolving, right? That's just one sort of thing that rises from these assumptions. But what genetic drift says, essentially, is that random events have a larger effect on smaller populations. If we're taking a subset of a marble, if we have a marble bag with 5 red and 5 blue, and we take a subset with replacement, it's very unlikely that we're going to get the same frequencies in and out. And then that compounds on the next generation. And so in these simulations that I set up, you can see like in a small population, allele frequencies change very quickly. Along the y-axis here is the frequency of a p allele. Along the x-axis is the generations, where every generation is just a subset of, or a subsample, excuse me, with replacement of the generation before. And so populations evolve very quickly with purely genetic drift in very small populations, where in large populations, you can go many, many, many generations with very little change. And that's just because, you know, if you have a million red and a million blue marbles in your bag and you pick a million with replacement, it's going to be somewhere near the middle. Okay, the next evolutionary force is mating system. This is on your ring finger, remember? And very simply, it says that if individuals with similar or different phenotypes preferentially mate, then those alleles involved in that similarity will rise in frequency. The example given here is if redheads only mate with other redheads, the redhead allele will rise in frequency. The third evolutionary force is mutation. I feel like mutation gets a pretty bad rap. You see like something, oh, that's a mutant. That's bad. But mutations are good, I promise. They occur in every single generation. You, me, everyone else here has mutations that your parents did not pass down to you. They're novel mutations inside of your genome. Every single one of us does. And importantly, those mutations, they accumulate over successive generations. That's very important for us as evolutionary biologists because I've given you a tree here on the right where I've shown you the different mutational steps, but when we go out and we sample, that's not what we get. We get this. We get only the contemporary samples, the samples that are alive today. And since we can rely on this accumulation of mutations over successive generations, we can say a lot about how these individuals are related simply based on what they are today. So in this example, the two individuals here on the left share the C allele, whereas the two individuals on the right share the G allele. We know that those are probably most similar. And you do this thousands of times across the entire genome, you get a pretty good idea. This is an aside, but it's important. Mutations are the only process, the only evolutionary force by which new diversity can arise. All the other forces just shuffle what's there, but mutations are the only way to get something new. And that's why they're so important. Okay, the next evolutionary force is gene flow. This is immigration and emigration, specifically of genes. And in my toy example here, we have a pond with 100% whitefish and 100% blackfish. And then over time, if you have migration from the blackfish pond into the whitefish pond, your allele frequencies will change, right? So in this, the whitefish allele is at 100%. In this, the whitefish allele is at 66%. Allele frequencies have changed with time, right? Pretty simple. The last evolutionary force is selection. There are many, many types of selection. The most well-known type is natural selection. This is, Darwin's stuff. And natural selection can be sort of subdivided into two components, survival and reproduction. Those are both necessary and neither one is sufficient, right? For evolution to occur, for allele frequencies to change, it doesn't matter if you survive if you don't reproduce. And on the flip side, it doesn't matter if you can reproduce really well if you don't survive to the age at which your species reproduces. And so natural selection is really these two things, survival and reproduction together to change allele frequencies. The example given here are just Darwin's finches, famous example with the different beak shapes and sizes, really small, really agile beaks here being really good for like berries and stuff, really large, robust beaks being really well used for cracking open nuts and eating the seeds. And depending on weather patterns of the islands, either nuts would do really well or berries would do really well. And so the birds that fed on those things would do really well and reproduce. Okay, so that was a quick primer on the evolutionary forces. I know that's not everything. I have studied this for quite a while. I know it's not everything, but let's return to Weedy rice, right? let's solve the mystery of how this happened, how we went from 0% resistance to 100% resistance just so quickly. So the first thing that I do when I come across a problem like this is I gather everything that we know. So what do we know? We know that, like I said, in just 16 years, we got to nearly 100% resistance. We know that weedy rice self-pollinates about 99% of the time. I think I forgot to mention this earlier, but basically self-pollination is the strictest form of inbreeding, where a plant will grow up and then its own pollen will land on its own stamen and produce the new fruit, the new seed. And we know from previous work that several mutations at ALS confer resistance to these imidazole herbicides. So this is what we knew coming into this study. And so we use these facts to sort of design experiments and plot a way forward. That way forward for us was whole genome sequencing of 48 plants that I collected from Arkansas. And whole genome sequencing is exactly what it sounds like. You start at the first nucleotide and you sequence until you get to the last nucleotide, and you can lay them all out on top of each other and see the different changes that occur. What I'm showing here, I know is a lot. You don't have to look at it too closely, but this is a very zoomed in view of ALS, of that gene. And it's what's called a multi-sequence alignment, where all of our samples are on the horizontal, and then they're aligned so that each nucleotide matches along the vertical. This green bar up here tells us that for every single one of our samples, this nucleotide matches all the way down. And where that deviates is where there are mutations. So let's look at those mutations and ask, do any of them change the amino acid? Because the central dogma of biology, transcription into translation, the actual genetic sequence doesn't matter. Changing the actual genetic sequence doesn't matter if you don't then change the amino acid. So we have to look for amino acid changes, what we call non-synonymous changes. And we found a bunch. Specifically, these two here are a G to A mutation at 1958 and a second G to A mutation at 1961. So what amino acids do they change? Well, they change some pretty well-known amino acids, serine 653 to asparagine and glycine 654 to glutamine. These are the mutations that are involved in Clearfield resistance. If we go and we look at the Clearfield cultivated plants, what mutations confer resistance in those plants? It's these mutations. And I've highlighted them here in red. You can see the Clearfield 161 has a N instead of an S. It has a asparagine instead of a serine here, conferring resistance. These other plants here, they have a glutamine E instead of a glycine G at the next mutation. So I'm sorry, the next amino acid. They're literally neighboring amino acids that change. But this should sort of confuse you a little bit because I told you that weedy rice is primarily selfing. How did these mutations get into the population if there's no gene flow, there's no interbreeding between the cultivated and weedy rice? And so to answer that question, then we move on to another analysis called a haplotype tree analysis. This is a lot here, but very simply, haplotype tree is just where we take genomes from every individual. We split the two chromosomes. They're diploids, so we split the two chromosomes, and then we those are called haplotypes. And then we look from nucleotide 1 to the last nucleotide, and we collapse all the individuals that are exactly identical. Um, so we take identical haplotypes and we combine them. The size of the circle is the number of samples that have that exact sequence from start to finish. Then we separate the circles by hash marks, where every one hash mark is one mutation. So for example, this individual here and this individual here are separated by 4 mutations. This individual here and this individual here are separated by just 1 mutation. And so I've then overlaid the level of resistance on top of this haplotype tree. So I've made it even more complicated. But what that shows us is we have this haplotype here, red, this is susceptible, and with just one single mutation, we get to the majority of our samples being highly resistant. And the same here. So we can ask, what were those mutations? And lo and behold, they are our known resistance mutations. This one here is our glycine to glutamate 654. This one over here, this is our serine to asparagine 161. These are the clear field mutations. The eagle-eyed among you might also notice that there's two more highly resistant haplotypes over here, and this gray one essentially means susceptible. And so another one mutation separates those. What happens there? And when we ask what mutation is represented by this hash mark, they're the same mutation. Here's glycine 654 to glutamate, here's serine 653 to asparagine. They're the same, but they're on opposite sides of the tree. That's weird. Why did that happen? So in order to understand this, we have to go one step further and return of the structure plots that I showed you before. Remember, one vertical bar is one sample. The degree to which it's colored is the degree to which structure has assigned to that population as being more like all the others of that population. The colors have changed, so let's go through it very quickly. Here in red is the US crop. This is tropical japonica. Here in purple is SH, and here in blue is BHA, black hole and. Everything that's about to pop up is those 48 samples that I collected. And they look weird, right? They look so weird. They are highly differently colored. They look nothing like the previous samples. And remember, these were collected in the 1990s before Clearfield. These were collected in the 2010s, long after Clearfield. And they kind of separate into these three groups. Here you have the purple and red group. There's green in there too. There's a reason for that. I just haven't gone over it. There's purple and red. These are SH by crop hybrids. We have this other group on the right here. This is the blue and red group. These are BHA by crop hybrids. This is super weird. 99% of the time they self. Why are most of our samples crop weed hybrids? The third group is this purple group here in the middle, and they look what structure is essentially telling us here is that these samples, there's four of them, these samples are indistinguishable. These four samples collected in 2018 are indistinguishable from these samples collected in 1990. And that's weird too, because these samples that collected in 2018, those were collected in fields that were spraying herbicides. Why does something that's genetically identical to the 1990 samples, why are they resistant to the things happening in 2018? How does that even happen? So these are essentially non-hybridized SH plants. So it's very strange that we found those. Okay, so now let's sort of go back a level. I said we had to understand structure to understand the haplotype tree. So let's go back to the haplotype tree and ask, where do these samples go? We know that there's SH hybrids, we know there's BHA hybrids, and we know that there's non-hybridized SH plants. So let's plot them out. That's weird too. Actually, it's not weird. It explains everything. We have over here, this block here in blue. These are all cropweed hybrid plants. All of these plants, remember what I'm showing you is the gene sequence of ALS. All of these plants got their ALS sequence from cultivated rice, from Clearfield. Gene flow moved the cultivated resistance allele from cultivated rice into weedy rice. And these plants over here, these are pure SH plants. They have the resistance mutation, but they did not get it from cultivated rice. They re-evolved it independently. So yeah, we have pure weed sequence and we have pure cultivated sequence in weedy rice plants. And that's just so cool, right? Because we ask if we look back to what our question was, how did this happen so quickly? And the answer is actually it took two different approaches. The first approach being grabbing the cultivated allele from resistant neighbors, and the second approach being mutating it ourselves, right? So that was so surprising to us, and that was just so cool to find. You don't ever find that, and we did. So I'm super happy. Uh, that's sort of the end of this story. So, getting back to our evolutionary forces, what can we learn? What did we use them for? How did that help us answer our questions? Well, finding crop alleles in weedy rice suggests that gene flow is the primary avenue of resistance evolution across the region. I mentioned that the size of the circle is the number of samples that have that particular haplotype, and you can see that this circle is just so much larger than these two circles, than any of the other circles here. The primary avenue of resistance evolution was crop weed hybridization. And what that suggests is that there's super strong natural selection for herbicide resistance. And this should be kind of self-evident, right? If you get sprayed with herbicides before you can reproduce, then that's the end of your genetic line. So being able to survive herbicides is super, it's easy to see why that would be natural selection. Um, then we have mutation also playing a part. The independent evolution of two resistance mutations was super cool to find. The figure I'm showing here, this is called local ancestry. And basically what we're asking the program to do is as we march across chromosome 2, here's nucleotide 1 of chromosome 2, here's the last nucleotide of chromosome 2. As we march across chromosome 2, we ask the program to tell us if that block belongs to cultivated or weedy ancestry. And when we, point out where ALS is, it's pretty easy to see that, it's marching along as weedy ancestry, and then all of a sudden we have this giant block of cultivated ancestry. Very strong natural selection for the cultivated allele here at ALS. Um, we took these data, and that was my PhD work. For my postdoc work, we extended those. I went to a bunch of different counties from southern Missouri to the coast of Louisiana here, and collected a ton more samples, and basically redid all these analyses and found a lot of the same stuff. But what else we found was that the weedy rice was acting kind of strange after this hybridization event. So if you'll recall, I told you that there was like 150 years where cultivated weedy and weedy rice were growing right next to each other without ever sharing their genes, without ever intermating, right? There had to have been a reason for that. And so now that weedy rice has gained resistance, that reason is back and it's still important. And so what we find is that a return to a selfing mating system. We hybridize once to get this cultivated allele. We return to selfing, and what it's doing is actually purging non-useful crop alleles elsewhere in the genome. That's what this figure down here at the bottom is showing. There's a lot here, but basically if you take some chunk of the genome, you ask what fraction of that chunk belongs to cultivated rice or weedy rice, you get some distribution here where the mean is well above zero point 0.5. Genetic drift suggests, because it's a large population, that it would stick around 0 0.5. It's significantly deviated from 0 0.5 here. And then this blue line is the chunk of the genome that contains ALS, and that is much more crop-like than you would expect just based on the genome. So this is a pretty busy figure, but basically what it's showing is that as weedy rice continues to evolve, it's starting to purge non-useful crop alleles. And this also makes sense, right? For cultivated rice, we want it to hold on to its seed. We want it to grow to a certain height. We want it to be a certain shape, all that kind of stuff, which isn't useful to weedy rice. Weedy rice wants to shatter, to disperse its seed. Weedy rice wants to not respond to the first drop of water. Because the first drop of water is going to be when the snow melts, right? And it's still way too cold. Um, we have all these things that weedy rice wants that cultivated rice. I'm sorry, we have all this stuff that cultivated rice wants that weedy rice doesn't want to do. And so you purge all those crop alleles, raising your fitness. And so those are our five. That's how we used the five forces of evolution to answer this question. And I'll just leave you here with this slide. The evolutionary forces guide evolutionary questions. If someone comes to me with sort of a proposal, a question they have, these are the five things that I'm thinking about. These are the five things that I can say, okay, well, this force and this force would predict this, but this force and this force might predict this other thing. So let's go look. Let's go design an experiment to particularly test one of these things, and let's look. Um, okay, so yeah, thank you so much. I love talking about weedy rice. Thank you to the Food and Agriculture Center for Science Education for letting me talk. I I love talking about my research. And I'd just like to thank my co-authors, Ken, Nilda, Tommy, Justin, and Connor, and then obviously my funding sources. So thank you so much.
Thanks so much, Marshall. I am thinking about our students in the high school or even middle school setting, and as we start thinking about this, it's like when we think about science and engineering prince or practices and students developing those skills, it's right here on their hand. They walk into the science lab and it's there on their hand for them to remember. So thank you for sharing that. We have 10 minutes set aside for questions. So if you have a question, if you go to the reaction portion on your Zoom and just raise your hand, we will open it up for you to ask questions. If you're not in a space where you can unmute and ask a question, feel free to drop it into the chat and we can read it out loud for Marshall to answer. Like to provide that good wait time as teachers and folks think.
Yeah, of course.
Jill, go ahead.
Hi, thank you so much for sharing your research. That was pretty amazing. I am actually a K-5 STEM teacher. I see all the kids at my school. And so my question is, do you ever do like a Zoom call with students, or would you be open to something like that?
Yeah, absolutely. My email is right there. Please do shoot me an email. Um, and I'd be happy to. Yeah, I love talking to students. I should mention I did at WashU we have this wonderful program. It's called the Young Scientist Program, where we as graduate students, it's a totally graduate student run program. We have a bunch of different programs that target K through 12 classrooms in St. Louis. And we have modules that we can do. We have all sorts of fun activities and stuff we can do. And I've been really involved in that for the decade that I've been here. And so yeah, I do. I love talking to students about science and about evolution and about biology as a whole.
Thank you. We have a school garden as well, and
Oh,
we've
fun.
seen some pests come in and take over. And so we've been
Uh-huh.
talking about, what we can do differently because we don't want to spray anything that would make it, a space where it's not okay to eat what we grow. And
Yeah.
so a lot of these conversations can, lead to what you're talking about as well. So thank you.
Yeah. And the goal, it's a little bit broader impactsy, but the goal of all this stuff, the reason you study herbicide resistance and stuff is so you can um, hopefully spray less herbicides, right? So we have this resistance to one herbicide. What's the natural comeback? The natural comeback is to develop a second herbicide resistant called strain of rice with a different resistance regime, a different resistance pattern. Spray that herbicide instead. And they'll, eventually, just like antibiotic resistance in bacterial and human health, eventually that sort of arms race gets to a point where it's no longer worth it. And so the reason that we study all this kind of stuff is to get a really good handle on sort of what went wrong, how can we mitigate it, what sort of techniques can we give farmers um, to really like prevent them from making scenarios in which herbicide resistance evolution would be possible, right? We had one farmer who admitted to us that he just planted this stuff every year because it worked until it didn't work. And we're like, yep, that's how resistance evolves. You just do it every single year. So really talking with farmers and studying these sort of the ways in which herbicide resistance evolves. The goal of it is eventually to spray less herbicides.
Thank you so much.
Yeah.
Is there anyone else who has questions? Feel free to just raise your hand. Oh, we have one in the chat. If the weedy rice self-pollinates, how did it acquire the resistance from the clear field? If you mentioned it, I apologize for missing.
Yeah, that's a great question. And I mentioned right at the end that there's been like 150 years of them all growing. We call it in sympatry, they're right next to each other, without any exchange of genes. And while that is true, it's sort of a lie. We do find um, every once in a while that there is like a hybrid. But the thing is that hybrid doesn't, it never survives. Uh, like I said, in that natural selection component, you must survive and you must reproduce. That uh, those hybrids, what they were doing was they were not surviving. They were forming because pollen movement is random, right? Like this plant isn't going to choose not to send pollen over here. Um, Pollen movement is random, so those hybrids were forming, but they weren't surviving. With this new like really really strong selection pressure where basically like the only things that would survive would be the ones that hybridized um we I think basically you killed all the weedy rice that didn't hybridize, and the only thing that did survive then was the ones that did hybridize. And I think it's just this like fluke event that happened. I should also mention that I said there's this system is very complex, as all systems are, and I mentioned that basically in every world region, weedy rice evolves from the local cultivated rice. The only place that's not true is actually here in the southern United States. We grow tropical japonica rice, but we have indica and Aus weedy rice. That difference, just the difference in strains from indica all the way to tropical japonica was enough that generally the hybrids didn't do well. Hybridizing two very different breeds of dogs, might not go very well. It's the same sort of thing here, is if you breed an indica plant with a tropical japonica plant, um, those offspring are not very fit. The thing that broke that, I mentioned in the structure plot that there's a green bit and there's a reason for the green bit. And it's actually because a lot of these clear fields, there's a whole nother level that I just never didn't get into. I didn't have time to get into for this talk. The things that are growing out there right now, the Clearfield varieties that are growing right now, are actually in and of themselves hybrids, and they're indica here in green, bitropical japonica here in red. And it's this inclusion of the green, it's this inclusion of the indica in the Clearfield hybrids that I think, and I have no evidence for this, but I think that it's this inclusion of the indica, the green bits here that allowed for those hybrids to survive. So very lots of layers. But yes, very well.
Great. Last call for questions. Okay, well, thank you so much, Marshall. We are going to move into uh, the next part, which is okay, Marshall shared this really, really cool stuff with us, this really interesting research. What does that mean for your classroom and bringing it into uh, your curriculum, bringing it in for your students? So how do we apply this research um, so that it can be an engaging and rigorous experience for our students? So I'm going to pass it on over to my co-facilitator, Angela, and she is going to share.
Thanks, Anna. In the chat, I just dropped a link for everyone. You should be able to access it. It will be view only, but you're welcome to make your own copy if you would like to save it in your drive. This is what we call a takeaway document. So anytime that you come to one of our virtual experiences, we want you to leave with all of the information that we feel would be helpful to you. So I think it was Leanne earlier that asked about, hey, are we going to have access to the recording? Yep. You'll have that. Plus, you also have all of these things, which will include, as you can see, the activity that we're going to do here in a moment. There's a link to Dr. Wedger's presentation, as well as several of his research papers and things that he shared with us and is willing to share with you as well. There's a video on there that talks a little bit about his research that he was kind enough to share. The Olson Lab research and links to all of the publications that are his and all of his colleagues. And then there's the Clover Lab research, which Marshall had mentioned in his conversation with Jill that, they have curriculum and things available and you can access some of that stuff on there. And then if you keep scrolling down, I did drop that link to the Five Fingers TED Talk, and we did put together a presentation summary in case you're like, wow, that was really high level and it was a lot. And it kind of did a little of this. At least for me, it did, not gonna lie. So if you need to settle back in, there's that. And then we also put together some other resources for you, and we'll share a lot about that later. But wanted to make sure you had that information ready and available for you. So again, one of the things that we wanted to do is make what Dr. Wedger and his team did in real life applicable to you all in your classrooms. And so obviously, Hardy-Weinberg is something that I remember doing this when I was teaching bio. So I think that if you are a biology teacher, you might be doing something similar or teaching all of these evolutionary forces and different principles in a different way. And so what we wanted to do is kind of utilize what Dr. Wedger and his team did and shared here. And we have some learning objectives that we've established for students related to that. So one would be applying Hardy-Weinberg assumptions to real world genetic scenarios, which I think the importance there is that real world concept because the phenomenon piece is so big and so important to science education. In that middle box, students will identify violations of equilibrium in that evolutionary process as the development of herbicide resistance has occurred within that population. And then the third box is being able to connect those evolutionary forces with agricultural impacts. So for us at the Food and Ag Center for Science Ed, it's really important for us to put together things that have true agricultural value and are real world agricultural problems and phenomena that students can explore. And so we're really proud to be able to share this with you. So we have two things that we're going to do. We'll spend some time here exploring a virtual version. We're not going to be able to finish said virtual version, but we wanted to give you a little bit of time and space to interact with one another and also with the content and the material as well. And then we do have an in-person version of the activity, which is set up more like a station rotation that's going to have clues, and then it's going to utilize some of the data and the research that Dr. Wedger shared with you during his presentation. So yeah, let's get to it. So on the takeaway document, the very first link that's on there is a link to a Google form. And when you get to said Google form, one moment here, sorry. When you get there, it will have 5 questions, and those 5 stations, kind of 5 puzzles that are set up, and each one is going to cover one of the assumptions of the Hardy-Weinberg principles that Dr. Wedger shared. And so we're not going to be able to get through the entire thing. If you are, like, more power to you, because that's pretty impressive. But each one of the virtual puzzles is going to have 1 to 2 to 3 questions. In order to advance to the next one, you do need to answer it accurately. And so hopefully we set everything up right for you to be able to experience it here for a little. To do this, we are going to send you into some breakout rooms so that you are able to chat with other people and all that type of stuff while you're in there. And it looks like we're getting some share requests, and that was what I was worried about. So let me go ahead and make sure that you are all able to access it properly because, why wouldn't we have done that before we got started, right? Um,
While Angela's doing that, our hope is that you kind of experience this from the student point of view. So you're going through this from the student lens, just being able to kind of see how students might engage with the Hardy Weinberg and the evolutionary principles that we shared. So we encourage you to talk, to go on camera if that works for you. And then when you come back, we'll kind of debrief and we'll talk about it from the teacher side of, hey, how does this, like, how might this fit in your class? And we have a non-virtual option as well. So we'll talk about that as well.
Thanks, Anna, for that. I am just changing that right now here. In the meantime, we'll get you into those rooms. And by the time you get there, you might have to refresh, but we'll take care of that right now.
Hey, folks, welcome back. We have about 30 seconds left in the room, so some people might still be finishing up. All right, and it looks like in about 1 second, everyone's going to be violently torn from that
A
room.
throwback. It's always my favorite in hosting these is when it's just a smash of like boom, 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 everybody popping back in. So welcome back. Thank you for kind of dipping your toes there into that virtual case of the rogue rice. Hopefully you were able to kind of see what our vision was of trying to take some of the complexities of what Dr. Wedger put together, and then be able to see how we might be able to utilize that in the classroom with students. So again, just highlighting that you had that virtual experience, but if you had a chance to look at the lesson plan as well, that 45-minute lesson plan is set up where it's kind of similar in the idea that there'll be like stations throughout the room, just like there were like puzzles that you had to go from one to the next. But there's also included in that a student handout that you can make a copy of and you can modify. We did also do some NGSS alignment so that you can see where it might fit into a classroom or where we anticipate it fitting. Particularly in this case, it would fit more in like an advanced middle school, high school bio kind of setting. But I know we have some friends on here today that are in those younger grades, and we'd happily work with you to adapt it if you needed some additional support in taking any of those that data or those questions and modifying that. So before we move on to wrapping things up for today, I just want to take a minute and ask a question of where might you see this kind of fitting in maybe what you already do, or maybe something that you think you might want to consider doing moving forward. So if anyone would like to share, you can drop it in the chat. You're welcome to come off mute since we're a small and friendly group. So yeah. Where might you be able to utilize this?
I'm thinking for me, I know it's higher level than kindergarten, but probably fifth grade genetics, trying to weave into what they do there. Um, and they really do like the um, inquiry based, like, here's the scenario, try to figure out what's going on type of thing. So this type of activity will work really well.
Love that. Thanks so much for sharing that, Jill. Yeah, one of the key components to what we consider to be high quality science instruction is this idea that it's around some real world phenomena. And for us, we again rely so much on agriculture because there's so much connection there for what students do every day and see every day and eat every day, and all of those things can be connected back to ag. So would anyone else like to share out where they might see this fitting in their classroom, or maybe just even a reaction to the experience or the resources? I know someone, at least one person's got to have a really good idea. Y'all came with great ideas. You came here on a Thursday afternoon in your summer vacation, so you all like learning. All right. Well, when I was teaching biology, used to set up stations that went through things like gene flow. And if all of you remember getting out like the little pony beads in the bottles and all of those different activities that you set up for these evolutionary forces, this could just be a way that you can introduce a real world concept. And again, a phenomenon based, like Jill said, very student driven experience, whether you're doing it virtually or in the classroom. These resources are yours to do whatever you would like to do with them. We also have plenty of other things as well available, and that's what Hannah is going to share with you here to wrap us up. So thanks again for coming today.
Yeah, so we have two things for you. We have other resources available from the Food and Agriculture Center for Science Education. That's our first link there. And then the second one is a survey link, just feedback on how we showed up today for this virtual forum, what you got from it, all that good stuff. So those are both there for you. I'll wait just a minute to allow you to be able to grab those with your phone. And the QR code scanner. And then while you're doing that, this is part of a series. This is the first in a series, and we'll be talking to more folks who are doing research connected to agriculture. So animal science, plant science, natural resource sciences, the things within agriculture that you can think of. And so we have a couple more folks lined up, and we're excited for y'all to join us. Our next event will be in August. And so you're on our email list because you came here. So we'll send that in our email list, and you can hit the register button again and register for our next event. It will be same setup. So we'll hear from the scientists about their research, and then we will be able to move into this kind of second half where we share some resources for you to be able to bring into your classroom. So we thank you so much for joining us. Thanks for connecting with us. And again, Dr. Wedger, thank you so much for sharing your research with us and making it so that it's a skill that walks around with me now. I might be sharing this with random people on the street. I don't know, but thank you. And we can't wait to see y'all next month. Take care.
Thank you.